Hello student, after the completion of the alphabets of principle of inheritance, now we are ready to take the chapter principle of inheritance. Now, so we begin with very initial time of initial time of genetics. So first we start an Australian monk whose name the Gregor John Mendel. What is the objective of Gregor John Mendel? The objective of the Gregor John Mendel is to observe how the character are transferred from generation to generation, from one generation to another generation, or say from parents to offspring. This is the objective of Gregor John Mendel. The question is for his observation, Mendel chose a Pisum sativum, is a normally we called pea plant. The question arises is why Mendel take the pea plant? Why not another plant like banyan tree or neem tree or mango tree? So the first reason is the availability of the pea plant. Availability of the pea plant. The question arises is it is the only thing the Mendel choose the pea plant? The answer is of course not. The certain another reason for choosing the Mendel of pea plant is contrasting trait. And what is the mean of contrasting trait? Contrasting trait, it means like two extreme character. None character is between them. If you take the height, like plant height, either you see a tall plant or a dwarf plant, none of the intermediate height plant. So the first reason for taking the pea plant, a Pisum sativum plant, is the pea have a contrasting trait. These traits such as pea have for height have a tall or dwarf. For seed shape either have round or wrinkled. For flower color have a violet color or white color. None is between them. इनके बीच में कोई और कैरेक्टर नहीं है, लाइक पिंक नहीं हो सकता और ब्लू नहीं हो सकता और कोई कलर नहीं, या तो वाइट होगा और या तो वायलेट होगा. So that's why Mendel choose the pea plant. And second reason for choosing the pea plant is short life span and hundreds of progeny. Short life span. If Mendel choose the banyan tree, the life span of banyan tree is hundreds year. So Mendel can see one generation of banyan tree in own whole life span. So they can't study the characteristic, the variations. That's why the Mendel choose the short life span plant. And he for knowing the characteristic of multiple generation of the pea plant. And third one is self-pollination. Mendel have the advantage of self-pollination is to create the pure line. For selfing, Mendel create a pure line family. And another is advantage have a cross pollination. By artificial hybridization, Mendel can perform the cross pollination in pea plant by emasculation. Okay, so initial time Mendel take 30 to 40 character, but at the end Mendel stuck 7 contrasting trait. Mendel give up on that character which so intermediate stage. Okay. At that time, at the end of his experiment, Mendel chose 7 contrasting traits, and these character, 7 contrasting character. Okay, not trade. Trade is different. Trade, if question is how many character Mendel takes in hybridization, the answer is 7 character. But how many contrasting trait Mendel is taken is 14. It's 7 is for dominant and 7 is for recessive. Okay, for 7 character, and these 7 character as follow, like seed shape, seed color, flower color, pod shape, pod color, flower position, and stem height. For each character, Mendel takes 2 traits. One is dominant, another is recessive. For seed shape, round is dominant. For seed color, yellow is dominant and green is recessive. For flower color, violet is dominant, white is recessive. For pod shape, inflant is dominant and flaccid are recessive. For pod color, green is dominant and yellow is recessive. For flower position, axial are dominant and terminal are recessive. For stem height, tall is dominant and dwarf is recessive. For methodology, how the Mendel performed the hybridization. For cross pollination, the methodology how the Mendel performed the cross is to take the pure line, to take the two pure line parent. The one is dominant for pure line, another is recessive for pure line. By choosing these two parents, Mendel performed this experiment. He take the two parent are pure line parent, pure line for tallness and pure line for dwarfness. Okay, and first Mendel do is cross pollination. After cross pollination, Mendel do the selfing. Okay, selfing in F1 generation. So what is the selfing? Like capital T cross with capital T, small t cross with small t, capital T small t cross with capital T small t. This is the selfing. When one parent is cross with identical genotype of the parents is called self cross. Or what is the cross pollination? When one parents cross with different different genotype of parents, this is the cross pollination. The first generation Mendel do the cross pollination. And why we call monohybrid cross? Mendel, each time of crossing, Mendel takes one character. It means when we study the plant height, we can't observe the similar like position, flower position, pod shape, pod color, pod shape, flower color, and other character. We only consider one character at one time. That's why this cross is called monohybrid cross. Okay, for monohybrid cross is first generation, Mendel take the homozygous dominant and homozygous recessive parent. By doing the cross pollination, first they produce the gamete. Okay, this is gamete, and the first F1 generation produced capital T small t. In F1 generation, Mendel observed all the F1 progeny are tall. None the in F1 generation, Mendel observed the the recessive character are not observed in F1 generation. In after the selfing in F1 generation, Mendel got F2 generation. Its second filial generation is F for filial. Is F1 is first generation or first filial generation, when F2 is second or second generation or second filial generation. In second filial generation, Mendel got capital T, capital T, capital T, capital T, small t, and small t, small t. Here, the phenotype, Mendel got 3 tall and 1 dwarf. Mendel got 3 tall and 1 dwarf. Okay, so what Mendel observed in F2 generation, he observed the recessive parent character are observed in F2 generation, which are hidden in the F1 generation. So this is the okay. Now in F2 generation, we got a 3 tall plant and 1 dwarf plant. Now the question is that ये जो 3 प्लांट हमें मिले हैं इन ये 3 प्लांट हमें मिले हैं ये F1 और F2 में, whether the tallness is due to capital T or capital T small t, because what Mendel can see, Mendel only can see the phenotype, is not genotype. That's why. So for knowing the dominance, for knowing the genotype of that F2 generation plant, Mendel performed the test cross. The test cross, the name suggests that कि हमें ऑर्गेनिज्म का जो हमें F1 और F2 में मिले हैं उनके जीनोटाइप को टेस्ट करना। तो टेस्ट क्रॉस कैसे करते हैं कि ऑर्गेनिज्म विद डोमिनेंट फिनोटाइप क्रॉस विद होमोजाइगस रेसेसिव इंडिविजुअल. Okay, so here is capital T cross with small t, then we get capital T small t 100% tall. It means none of the plant show the dwarfness. So the plant which we take so are homozygous dominant, showing the homozygous dominant. When we take capital T small t cross with small t, then we get 50% of tall and 50% of dwarf. That 50% of tall and 50% of dwarf is the 50% of tallness or 50% of dwarfness is showing here which plant we take. The genotype are capital T small t. It means which plant we take are not a homozygous, it's a heterozygous. One thing you are notice here in F test cross, the genotypic and phenotypic ratio are same. It means the phenotypic ratio is 1:2:1, then genotypic ratio is also 1:2:1. On the basis of monohybrid cross, Mendel proposed two laws, the law of dominance and law of purity of gamete. Law of dominance, what the conclusion of monohybrid cross. Conclusion of monohybrid cross is law of dominance. Okay, the law of dominance have three propositions. Mendel proposed the three proposition of law of dominance. The first is every character is controlled by a discrete unit called factor. It means every character. It means if a gene is controlled, the tallness is control. Sorry, if a gene is for tallness, he or that gene, sorry, if a gene control the tallness, that gene cannot affect the other character like seed shape or seed color or flower color. Only a gene is a वो जीन जो टॉलनेस के लिए इफेक्टिव है वो किसी और कैरेक्टर को अफेक्ट नहीं करेगा, वो किसी और कैरेक्टर को शो नहीं करेगा. अगर एक कैपिटल टी टॉलनेस को शो कर रहा है तो वो और किसी कैरेक्टर को लाइक सीड शेप को, फ्लावर कलर को नहीं शो कर सकता. ये फर्स्ट प्रपोजिशन का कहना है. सेकेंड प्रपोजिशन इज सेड्स एवरी कैरेक्टर अकर्स इन अ पेयर. एवरी कैरेक्टर अकर्स इन अ पेयर, ईच कैरेक्टर पेयर में अफेक्ट होते हैं. लाइक टॉलनेस, tall is character, okay, but sorry, height is character, but tall and dwarf are trait. Okay, the tall have capital T, it means a factor is trait or tallness, and that tallness is controlled by factor which are in pair, and that factor is capital T or capital T small t. And third proposition is, in a pair of dissimilar factor, the one that expresses itself in both homozygous or heterozygous condition are dominant, and those who express itself only in a homozygous condition, that is called recessive one. And here is some exception of law of, here some exception in law of dominance have. The first one is pleiotropy, multiple allelism, incomplete dominance and codominance. Let's discuss one by one. For the first proposition, the pleiotropy, first proposition is says that the every character is controlled by a discrete unit is factor. The exception is pleiotropy. What is pleiotropy? Pleiotropy says the single gene can affect more than one phenotypic expression. Single gene can affect more than one phenotypic. Like for example of pleiotropy is starch synthesis in P. The gene which affect the starch synthesis in P also affect the size of the P, the size of the P plant, the shape of the size of starch synthesis, starch grain and shape of the seed of the pea plant. Another is accumulation of phenylketonuria, the association of phenylketonuria. The accumulation of phenylalanine cause three characters, like one is mental retardation, second is pigmentation in skin, and pigmentation is here. Here we can say a single gene can affect more than one character. One character is mental retardation, second is hair pigmentation and skin pigmentation. Here we also say as this is a pleiotropy. And second one is every factor occurs in a pair. The exception of that second proposition is multiple allelism. What multiple allelism say? For a single gene, more than one allele are present. The example are ABO blood group system. In human have a IA. For human ABO blood group have three allele: IA, IB, and I, small I, I naught. And third proposition is third proposition is that in a pair of dissimilar factor, that one expressed in both homozygous or heterozygous are dominant, and which expressed in homozygous condition is recessive. But here is exception are incomplete dominance. Where we see such incomplete dominance is flower color in snapdragon. In snapdragon, we taken here a parent of capital T, capital R for red, and white for small r, small r. The gamete are capital R and small r. What we get in F1 generation is pink flower. What we get in generation is pink flower. So but ideally according to Mendelian hybridization, यहाँ पे क्या होना चाहिए, यहाँ पे रेड कलर शो होना चाहिए, जो कि यहाँ पे डोमिनेंट क्या है, रेड कलर डोमिनेंट पेरेंट्स को शो कर रहा है, और यहाँ पे वो डोमिनेंट कलर रेड होना चाहिए, जबकि यहाँ पे पिंक है. सो देयर वाज अ सरप्राइज how the particular thing are came, so this is why we call the incomplete dominance. And the incomplete dominance, another example are codominance. Let's be discussing some more thing in codominance or incomplete dominance. When we cross the F2, when we cross these F1 plant, F1 generation plant, when we do the selfing, we get capital T, capital R, capital R, capital R, small r, small r, small r, sorry, capital R, small r, and small r, small r. And what we get here are one red flower, two pink flower, and one white flower. So the F2 generation are identical to the Mendelian character. Mendel in F2 generation, Mendel in ideal Mendelian hybridization shows similar type of example, similar type of this